built in a little bit of extra time at the end. So please take your time. And Sam is the head of patient policy affairs for Lysogene. I'm sure many of you already have met her and know her from previous meetings, and we're looking forward to her discussion today. Thanks, Sam.
Thanks very much, Doug. Great to be here. Very strange being in a virtual meeting with everybody. I would much prefer to have a face-to-face and be talking with you and meeting your children. But nevertheless, having just heard some of the presentations, I think there's one thing also that I'd like to say is it's so exciting that there are so many research programs and particularly gene therapy programs in GM1. I think this is great for the community. And to know that all of us working, whether it be in research, academia, and in companies, we are acutely aware of the necessity to collaborate and put the family first. And I think that's what we will all do. So it's really a great thing, and a great thing to hear my colleagues presenting other gene therapy programs. So I'm trying to pass the next slide and it's not happening.
Samantha, just click on your slide first, and then you should be able to click on the spacebar arrows.
Oh, wonderful. That made
Perfect.
me nervous. Thank you. This is my disclaimer. And how do I need to close the thing so that I can actually read my slides? Let me do that. There we go. So yes, no, I wanted to Lysogene. So Lysogene as well is a company, it's a biotech company that was founded in 2009 by a lady called Karen Ayash. And her child was also affected by a neurodegenerative disease, a disease a little bit similar to GM1. And so the culture of Lysogene is very much rooted in a deep and compassionate understanding of the impact that these diseases have on patients and families. And we're working very hard today to develop a potential treatment for GM1 really as quickly as possible. And obviously on the left here, we have some beautiful photos that I would like to thank Cure GM1 Foundation for having shared. So just to explain, and earlier, I think there's always been a great description, but just to go through again, what's causing GM1 gangliosidosis. So here we have a normal, so this is, in this case, it's a normal nerve cell with a working gene, so a working GLB1 gene. And that gene is able to produce this enzyme called beta-galactosidase, the enzyme. And this enzyme is able to break down GM1 ganglioside. And that happens inside the cell. What happens when we have a cell, in this case, again, a nerve cell with a faulty GLB1 gene? It means that that enzyme is either not working properly or not produced at all. And then it's not able to break down the GM1 ganglioside. And therefore we have a storage, we have a buildup of this GM1 ganglioside that causes severe damage, mainly in the brain and spinal cord. So at Lysogene, we've just discussed an intracisternal, so it's a very popular route of administration, obviously for GM1, and it's clear the reasons have been very, very well explained earlier, why we're using an approach like this. And I'll go on also to explain a little bit more about the other approaches that we have looked at and compared in the animal models. So the goal of Lysogene's gene therapy is to deliver a working GLB1 gene into the cells of the body to enable them to make the enzyme. Since the most damaging effects of GM1 occur in the brain and spinal cord, it's critical that enough therapy reaches those cells. A modified virus, so here we have the virus, the vector and the GLB1 gene. So this virus is carrying the GLB1 gene, is delivered directly into the fluid that surrounds the brain and spinal cord in this area that was described again earlier, called the cisterna magna, that's at the back of the head. Injecting into this area helps to get as much therapy as possible to the nerve cells. And the actual injection takes about 5 to 15 minutes, and the child is asleep all the time. And it's expected that this missing enzyme will be made and released throughout the brain and spinal cord, and as also was described earlier, to the peripheral organs. So this slide presents some of the preclinical work. So we've been working with for some years on the preclinical in GM1, And I'm very happy to hear also to describe some of the nice collaborative work that happens, because in the work that we've done on the mouse model, we've worked with Miguel Esteves and Doug, you know, our cat works. So if we have any questions on the cat, Doug's the man. So what we've initially what we did when we started working with Doug and Miguel is looked at different vector constructs. And here a decision was made to work with the AAV Rhesus 10, so it's one of the AAVs, and it's actually, Lysogene is a phase 3 company. We're already doing a late stage gene therapy program with the same vector, and obviously here we're using the gene of interest for GM1. And so we've looked at the mouse model, and if you see on the right here, so we've looked at a knockout mouse of GM1, and we've looked at the enzyme activity when you treat the mice, and we've also looked at the impact of that enzyme activity on the GM1 ganglioside accumulation. So on this bar here, so this shows a normal mouse, a wild type mouse, and this bar here shows a mouse with GM1, so the knockout mouse with GM1. And here we have 3 different doses that have all been tested in the mice, and we've looked at different parts of the central nervous system. And here we see that there's a significant increase in enzyme activity, and that's producing a long-term activity and a reduction in the GM1 ganglioside accumulation. We've also looked at, which I don't have pictures here, and done this work with Doug Martin, and here we've looked at different routes of administration, and of particular interest, and there's different research groups doing different things. It's all investigational at this time, and that's a good thing. And we've looked, our particular interest is delivering into the CNS. And so we've looked at intracisternal. We've also looked at, I think people can see my photo, intracerebroventricular, which is another way of being able to deliver into the CSF. And we've also looked at intrathecal, where there's just an injection, similar to a lumbar puncture into the spinal cord. And the reason for choosing intracisternal is that it's giving the best results, the highest enzyme expression, and also reduction in storage. We've also looked at the monkey, and the monkey is the model, particularly in gene therapy. It's a larger animal, it's closer to human. When you're looking at distribution, it's a very informative model. Obviously the monkey does not have GM1, so it's a normal monkey. And here you can see what happens with a normal monkey, and you're looking at the enzyme activity. And then we've looked at 2 different doses, and you can see that this increase is the increased enzyme activity compared to normal. And so that's a little bit of results from our monkey study. Also of point, I think it came up in the questions last time about the intracisternal route of administration. It is something that's new, because gene therapy is new. Gene therapy is a one-time administration. So you can have more invasive routes of administration when it's one time, and it's really something that's of great interest. And therefore, because gene therapy is advancing, there are already at least four, I haven't looked in the last few weeks, clinical trials in different diseases ongoing using an intracisternal delivery. So this is the clinical trial that we're looking at. It's an adaptive design. So we're trying to develop one longer study, which will include a significant amount of patients for a rare disease, about 18. We are already visible on clinical trials, but we're not yet recruiting. Our intention is to be recruiting this year. We will follow the children up for 24 months. And then on a longer term follow-up study. And we will have sites both in Europe and the US. And, as experienced from our MPS 3A clinical trial that we're doing, we have families coming from all over the world. That's something that we know how to do. I forgot to mention the slide before, but one of the things that we're also doing in the gene therapy trial is families will also be given the choice of recording videos. We mentioned a little bit of videos on the natural history because of COVID, but it's not because of COVID. And I'd like to go into a little bit more detail, the rationale of why we think video with or without COVID is a really important outcome to be looking at in these particular diseases. So this rationale is really based on, it was based on some research that we've also done in Sanfilippo MPS IIIA, the disease I was discussing before, that we're also doing video outcome. And there's been a lot of discussion and also talking to GM1 families. And what we know about, particularly, I mean, all rare pediatric diseases, but particularly in neurodegenerative diseases, is that, kids have, are coming in and into the clinic. They're not to the best of their performance. They're not performing the way they do at home. And there's several reasons for this. One of them is the travel. So again, I've just discussed that they will be taking children from many countries to travel to sites, and sometimes they have jet lag, or they've been traveling for a long time. They have travel fatigue, and then they arrive into the clinical site, and we're looking sometimes 2-3 days of different assessments. So there's clinic fatigue of just the number of tests and prodding about that's being done. Anxiety of the parents will also come onto the children, and just being in that environment, and whether it be a natural history study or particularly moving into an interventional trial creates anxiety. And then there's different people in a clinic. I know certainly my daughter does not like going to see the doctor, but even here, there may be language differences as well. So it's very impressive, I think, for the family as a unit, but particularly also for the child. And then there's also the difference about what a child will do when they're pushed, so a clinical performance, and what they will naturally do without being prompted. And this is much more reflective on everyday life. So this is one aspect of why we think that video recording, if it's done in a very rigorous manner, can be an important outcome. Another rationale for this as well is how important it is to really understand where the patient, where the family, where the child is in their disease journey, to be able to understand what are our expectations of treatment outcome. So this is just a typical curve, which is called a bell-shaped curve. And what you see here, it could be any outcome. It could be walking, it could be talking, it could be cognition. But in our disease, in this disease, we see that the children sometimes may be born and progress relatively normally or slightly off normal, but acquire new skills, and then they may reach a stage where that plateaus, and then there's a loss of skills. Now, the way we look at outcomes, for example, if you look at language, and in a child that's completely non-verbal, the value of being acquiring, again, speech is so much more than if you're looking at a child that's in the slope of development, is continuing to acquire new skills. And this is, again, something that we're looking at through the video. We can capture, and we also, because we've tagged onto this, interviews with the parents, and understanding their perspectives, their expectations, and what they're seeing. So understanding what they're seeing, what are they looking out for in their child. So these videos have been, I don't know if the volume, I do hope the videos will work. These videos have kindly been shared with me, and they're part of a video project that we're doing on the MPS IIIA by Cara O'Neill from the Cure Sanfilippo Foundation. So I hope everybody can see the video. So here we have a very important activity, and it's an activity that would be important to look at in GM1 as well. And this activity is mealtime. So we can see a child eating here, and this child is able to sit at the table and eat with the family, and eat relatively independently with help from, in this case, we can see help from dad. And so that's something that we can capture. Now we look at this little girl. This little girl's also eating, but she has the ability of being able to hold a fork, hold that fork and hold it properly, prod her food, and also put that food into her mouth. And we see a little bit later in the video that I'm going to stop the videos because I have the background noise, but we see a little bit later in that video that the mum helps her a little bit. Now, on the questionnaires that we use in the clinic, we would tick exactly the same box. We would be saying that that child eats with a little bit of help from mum or dad. So the importance of video and the difference that we can see in those two children, as well as just looking at that aspect of the disease. There's other things that we're able to see. We're able not only to see the self-feeding skills, but also movement, the fluidity of the upper limb movement, and we're also able to see the attention of the child during this moment, which for most children is a highly motivating task. Oops. So with all that in mind, we have worked, and I would like to give an enormous thanks to NTSAD and the Cure GM1 Foundation. And I work with a lady from Casimir. She actually has a son also with DMD, and the idea of these videos has really come from her personal experience in clinical trials. And this is the dream team. She's absolutely a wonderful and insightful and smart person to work with. And she's really, really developed this idea. And I know she not only works on this as an outcome, but her work is also towards validating and working with the regulatory authorities to take this one step further. So with this group, we've set this up in GM1, and it's, as I said, it's part of the, it will be part of the gene therapy trial where families can and also collect this. It clearly is, I hope you've seen from the slides before, you can see that what we believe is really going to enable a deeper understanding of the real world impact of GM1. It's a very rigorous capture because this is the result. And again, I'd like to thank, I think, I saw some, quite a few families, and I'm sure some of you have actually helped in designing what would be the hallmarks and what we need to be watching out for in GM1. And really, you know, coming from the family perspective, the people that live with the disease and understand the disease most. So it's been very rigorous, and there were many interviews that were done with parents of the different GM1 subtypes. These videos are independently reviewed by experts, so experts in GM1, but also we think, as we think about how we design that, it can be experts in movement disorder to really correctly be able to see change over time. And as was described by a colleague, these COVID just happened to us. We're working on another trial where we don't have just video, we also have the clinical visits, and it's quite challenging. Here, it's very easy. In fact, hopefully, the one advantage of COVID is we all have a little bit more time with our families. So as well as having that on the, as part, it will be part of the gene therapy trial, we've also set this up as a natural history. It's called the GEMEO, GEMEO, because the children are gems. And this natural history study is up and running. It's currently recruiting. We have already recruited the first families into this. It will certainly, because as I said earlier as well on my slide about the clinical trial, and has been the discussions throughout this meeting, talking about clinical trials in general and the feasibility. Do we want, for gene therapy trials, where we have a one-time therapy, a relatively invasive delivery, randomized controlled trials seem a particularly difficult option. And I know that the different regulatory authorities are particularly understanding of that, and also the understanding of this rare disease where it would be very, very difficult to recruit into control groups. So we're using that natural history that's being done as the control group, and this will strengthen as well that natural history. So at the bottom here, these different assessments. were designed with the families. As I said, those are the things that family described as they were particularly watching out for with their children. And then there's obviously a manual of how to collect that data. And again, one of the great advantages of technology today is that all the videos that you took many years ago, well, they have a date on them. And so we're able to collect retrospective data, and it's not, oftentimes retrospective data has been criticized because all our memories fail us to some point. You know, we think something happened in 2008 and oh my goodness, it was 1996. But a little bit of an exaggeration, but our memories fail us. And here we don't need our memory because we have a date on the video. And so the value of retrospective data, I think, is excellent. So if you are not aware of the study, I invite you to contact, so that email address here, GM1 @ Trials, goes directly to Mindy Leffler. As I've described earlier, maybe she's listening to this, is a wonderful person. I encourage anybody who would like to reach out to please do so. And I'm sure that Sue and the NTSAD team will be very helpful and able to direct you in the right place if you would like to join that study. And with that, I've presented, I think, a little bit about Lysogene, our preclinical, our clinical trial that's coming up, and our video study. But I'd particularly like to thank you, the children, the parents, the families, for your continued contribution, because it's these types of meetings where the research happens, and it gives us as well the energy to go back and just try and get treatments progressing quickly. So thank you very much. And if there's any questions.
Sam, fantastic. And I can tell you've done this before because you are right on time. You've got about 3 minutes for questions. So I have a question for you. You mentioned that the clinical trial is listed on clinicaltrials.gov, but you're not recruiting yet. Any idea when you'll start recruiting?
I can't give any more details at the moment, other than we anticipate this year to be recruiting into the clinical trial.
That is really exciting. And this is a question, sort of a carryover from the previous talk, but I think it's relevant here. And you did touch on it. You've decided to use the RH10 capsid, or the RH10 serotype, for the clinical trial. And you mentioned that you've used that in another trial, but maybe you could give a little bit more detail on how things have gone in that previous trial and why you chose to use RH10 in the GM1 trial.
Okay, I'm at Lysogene. I'm not sure I'm the most versed and best person. It would be our chief scientific officer. But I think having so I've been with Lysogene for 6 years now, and I think 6 years ago there was a lot of discussion about should it be. There was a lot of arguments around AAV 10 and 9 being superior to some of the older vectors 2 and 5, and I think that is absolutely true and valued today. However, I mean, in my humble and more lay opinion, in terms of that particular aspect of the question, what we see in 9 and 10 is pretty similar in terms of distribution, and pretty similar. I mean, in our case, I mean, a lot of the discussion is about, if you deliver it intravenously, does it cross the blood-brain barrier? Since we're delivering intracisternally, that question of crossing the blood-brain barrier is less of a problem, I would say. But what we see between those two vectors is pretty similar distribution, and good distribution, much better than the older vectors.
Yeah, I think that's really fair and a great answer. I thought, from the parents' perspective, they're seeing AAV9, AAV-RH10, and different serotypes, and I thought it would be helpful to clarify that a little bit, so thank you. We have a question from Nandini Malati. What is the name of the trial in clinicaltrials.gov, please? And
I don't think it has a name. So if you look, and when he has disease, you put GM1 or GM1 Gangliosidosis, and then in the row below put Lysogene, and you'll find both the natural history and the gene therapy trial.
Right. So the clinicaltrials.gov website has a search feature that you can just go in and search, as Sam just mentioned, and it will come up. And that's really exciting to see the clinical trials start to pop up on clinicaltrials.gov. It's amazing, Sam, isn't it? As we progressed over the years, to see these things finally coming to fruition and existence is fantastic.